Hey everybody, sorry I'm a bit late. My previous class was taking a midterm, and so I had to stick around until the end for that. All right, so good afternoon, everyone. How's everyone doing today?
Good. How are you?
I'm good. So I'm back in my office for today. It is hot today, yes, but I'm back in my office because I think the heat got to Spectrum, and so my internet died for like 3 hours in my apartment. So because I had to lecture and because I had an exam to give, I drove over to my office because I was hoping that the internet was working here. Yeah. So if you haven't come to campus, the way it works is that you have to apply for like a single day pass or something like that. And so I was filling out the pass on my phone while getting dressed, and I was just hoping that Dean Susan would see it before I got to campus, and she did. And so she approved it, and so I'm here, and I am approved, and I'll be here until the end of today. Okay. And so the plan for today is, I know there's been a lot of questions on homework 4, specifically problems 3 and 4, which is the recursion stuff. And so I'm going to spend the first maybe 20-25 minutes going over that, just to make sure you guys get some help with that. And then we have another set of lecture notes on microcirculation. Okay, and so that one is fairly short. There's not much for me to say about microcirculation. So I think the timing is good that we can spend some time talking about the homework today before you guys turn it in tomorrow. Okay. All right. So thankfully, for the homework, there's not that much code to write. I think it's more just the conceptual stuff of getting that down. And then once you have that down, then then you can kind of turn it in from there. Okay, so are there any questions before we get started today? Sorry, just dealing with the last few questions from the exam. Hang on, give me 2 minutes. Sorry about this, guys.
You're all good.
Okay. All right. So let's go ahead and jump back into let's look at the homework. Sorry, brain is frazzled right now. Okay. All right. And so the homework, basically, I'm asking you guys to implement a way to compute the both the resistance and the impedance of a vascular tree, right? So actually, let me go ahead and open up the homework document. Okay. Right. And so we're looking at a situation like this where we have a splitting tree and it goes just like that. Right? So let me pull up the starter code that I sent you guys. Okay. And starter code. Okay. Yes. Students on one end, starter code on another one. Okay. And so the starter code basically, this is the solution. All right. Right. We worked through this together in class. I think on a previous day. And so If you're having problems with problem 3, you can look at a previous set of lecture notes, and I think I go over that. I don't remember exactly which day it was, but I think we covered it at the very beginning of the class. And so if you look back at that day, then you should be able to get it from there. Okay, and so question or problem 4, I want you to repeat this this exercise, but with impedance. Okay, and so we're going to basically what I want you guys to do is to basically follow this this kind of algorithm right here for the procedure for solving for a system of bifurcations. Okay, and so this is the end at the very end of lecture notes 10. For finding this guy. Okay, and so the steps I've also outlined for you in the comments for this as well. Okay, and so if you open up the starter code for the impedance one, which is problem 4, I've listed basically all the steps that you need to do. So let's quickly just kind of go through each of these steps, and I'll show you kind of the code that you need to write for each one. Okay, all right. And so, I think with these kinds of problems, I think it's easy to get kind of intimidated and kind of lost at kind of like the overall, task that you have to do. But, if you kind of break this down kind of step by step, then, each step individually is not so bad. Okay, and so let's start with the first step here, which is computing the characteristic impedance of the vessel segment. Okay. All right, and so the characteristic impedance is given by this this formula right here, right? And so characteristic impedance, we need rho, we need the speed of the wave speed c naught squared, okay, times lambda divided by I omega a, okay. And so the two variables that we need to compute here first are going to be lambda and a, right? And so let's first compute a, because a is kind of the easy thing to compute, okay. And so a is simply just going to be pi times radius squared, right? Because remember, we're assuming that we have a circular cross-section vessel, okay? But for the radius, remember, the radius is going to depend on what order our vessel is, okay? And so we're going to have pi times vessel radius at order squared, okay? All right, and so that's the area, and then that's the one that we can use for that, okay? And so the next thing that we need to compute here is lambda, okay? And remember, lambda is this kind of this expression that we have here. And so if you want to know kind of the exact definition of lambda, it's basically the one of the eigenvalues of this solution, okay? Question, what does order mean, right? And so order basically is going to tell you the size of the vessel, right? And so what this problem tells you is that you're going to start at a vessel of some order. And so actually, I think for this problem, we start with an order of 11, and then every time it bifurcates, the order of the two daughter vessels is going to go down by 1. Okay, and so just as an example, let's say that this guy is order 11. Okay, and then when it bifurcates, it's going to produce one of order 10, and then another daughter of order 10. Okay. And so the idea with the order is just to kind of emulate the fact that when a vessel bifurcates, it goes down in size, right? So remember when we talked about the very beginning of class, when we talked about Murray's law, we said that whenever a vessel bifurcates, it goes down in size, right? And so kind of like what we talked about when we talked about problem 3 of the homework, I've listed out all the different sizes in terms of length and in terms of radius for those different vessel orders, okay? And so the way to kind of read this is that, at the start here, this is a vessel of order 1. And so a vessel of order 1 will have a length of 0.005 centimeters and a radius of 0.00133 centimeters, okay? And if you look at the other end of the spectrum, a vessel of order 11, which is the end of this vector here, is going to have a radius of 0.1639 centimeters and a length of 1.811 centimeters, okay? And so when we compute the area of the vessel, we want to do it for this respective order. And the order is loaded in as a parameter to this function. Okay? And so when you call this function, the function is going to know what order it's going to be. Right? Okay. All right. Okay, and so that's the area, and let me check with the solution here, okay? Yes, and that is what we get for the area, right? Okay, so the next thing that we need to compute here is lambda, okay? In particular, I'm gonna compute lambda squared, okay? And so let's go ahead and look at the formula for that. Okay, and so lambda squared is going to be this formula right here. Okay, and so we need to do 1 divided by the wave speed squared times parentheses minus omega squared plus I omega 8 pi nu over a naught. Okay. Okay, and so let's go ahead and put that formula in. Okay, and so we have lambda 2. Okay, and let me just because the student is still messaging me, let me go ahead and just copy this this formula right here. Okay. And so lambda 2 will just be 1 divided by the wave speed squared times omega here is frequency, okay? And remember, frequency is another argument for the function up here, okay? And so we have 1 divided by the wave speed squared times minus frequency squared plus 1i times frequency times 8 times pi times viscosity divided by a, okay? And so I basically just copied down this formula here for the lambda, okay? Okay, and so now that we have the area and we have the lambda, we can compute the characteristic impedance of this vessel. Okay, all right, and so let's see. Sorry, Isaac, your question. I see. You would have to define what the frequency is. So the frequency is loaded as another parameter in here as well. And so that's given to you in this function. And so the way that I've kind of structured this code is that, we're going to compute the impedance kind of one frequency at a time. And that frequency is given to you in this other function over here. Okay. Right. And so you can see here that I'm calling this function. You don't have to modify this code. And so you don't have to modify this function called, or this code called generate bifurcating tree impedance. And so, all you have to do is just call this function. But you can see here that I'm loading in this frequency in with the function, and then calling it from there. And so all you have to do is just reference this frequency variable that's up here. Okay. And the wave speed, I think, is also defined for you. Yes, the wave speed here is 200 centimeters per second. Yes. Mm-hmm. Okay, and so now that we have lambda 2 or lambda squared defined, we can go ahead and compute this characteristic impedance right here. Okay, and so the characteristic impedance, and so I'll call this Z naught, is equal to density, which is rho, okay, times wave speed squared, okay, times lambda. And so remember, we just computed lambda squared, and so we need to square root of lambda 2, okay? And so that's going to give us lambda. And we're going to divide this by 1i times frequency times a, which is what we computed there, okay? All right, and so that's it for the first part of this code. And so the first part of this code, we're just computing the characteristic impedance. And so we're just going to take this kind of one step at a time, okay? Okay, so any questions on this so far?
And then, Professor, we don't need to convert it as a complex. That's right. You just wrote I times frequency times 8 pi viscosity. Yeah, that
Correct.
Yeah.
Correct. Yes. Yeah. So any imaginary numbers, you just leave it as I, and then it'll deal with it later. So part of impedance is that you're going to get some complex numbers, some imaginary numbers, but that's okay. And so that's part of the impedance, and that's part of the reason why we can only do this with code, because doing this by hand would be really complicated. So yeah, any I, you just leave it there, and you don't have to worry about rationalizing or anything like that. Yeah.
Thank you.
Mm-hmm. Okay. All right. And so now that we've computed the characteristic impedance, we're going to use this in other parts of the calculations, right? Because remember, the goal that we want is that we want to ultimately compute the not only the reflection coefficient, but also the terminal impedance as well. Okay. And all that is going to use this characteristic impedance right here. Okay. Okay. And so that's kind of step one for this algorithm. And so step two is we're going to implement our base case. Okay. And so for our base case, we're going to, remember our base case is when the order is equal to 1. Okay. So if order is equal to 1, that means we've reached the end of the tree. So if we reach the end of the tree, that means we can go ahead and implement our base case. And so if we go back to the homework document right here, okay, let's see. Wait, where did I say this? Oh, is it in the code? Oh, it's in the code right here. Okay. So in the base case, we're going to have a reflection coefficient of 1. Okay. And so, remember, the first thing we have to do when we define our base case is we have to do an if statement, and then we have to define our reflection coefficient. And so we'll define a variable called reflection coefficient, and then we'll define it equal to 1. Okay. All right. And so with the reflection coefficient defined, now we're ready to compute the input impedance of this vessel. Okay. And so we're just following along with this kind of algorithm here. Okay. And so we've done step 2, where step 2 here is we have our reflection coefficient at the very least, for our base case. And so what we can do from here is we can just use that reflection coefficient to compute the input impedance of this vessel. Okay. And so to get the input impedance, we have to go back up to this formula right here. Okay. And we can see that input impedance is equal to characteristic impedance times 1 minus reflection coefficient times exponential minus 2 lambda 1 times L1, okay, divided by 1 plus gamma 1 e to the 2 lambda 1 L1, okay? All right, and so we can go ahead and input that formula here, okay? And so the input impedance here, we want to store that into this variable here called impedance. And so impedance, we're just going to implement this exact formula right here. Okay. Okay. And so we're going to implement this, and so we're going to have z0 times 1 minus reflection coefficient. Okay. Times exponential of minus 2 times lambda, which we compute earlier. Okay. And so lambda here is going to be square root of lambda 2, okay, times L. And actually, let me make another variable for L here just to make things convenient. Okay? Because L here, the length of the vessel is going to depend on the vessel order. And so L here is going to be vessel length at order. Okay. So do times L. Okay. Okay. And then we're going to do divided by. And just to make sure that we just to kind of save space here, I'm going to go on to the next line. And so we're going to have the exact same thing, but we're going to have 1 plus this. Okay, so let me go ahead and copy this. I think I forgot parentheses. Yes, yes, okay. Okay. And we're going to add a plus right here. Okay. All right, and so that's our impedance. Did I spell it wrong? Okay. And so that's our impedance. And so in that case, we're done. And so this is the case when we are on the base case, okay? And so when we're on the base case, we're able to compute the input impedance just based on the terminal impedance, as well as the given, or I'm sorry, we're able to compute the input impedance based on the characteristic impedance Z0 and the given reflection coefficient of 1. Okay. Okay. So any questions on this? Sorry, this is driving me nuts. Okay, muted for 8 hours. Okay. All right, and so that's the base case. And so the base case is when our order is equal to 1. Okay. And so we've taken care of that. And so for the case when our order is 1, and our reflection coefficient is given, then we're able to, compute the input impedance and return from the function. Okay. And so if you compare this to question 3, right? And so this is basically the exact same thing for when we reach the end of the tree, and the resistance that we compute is just for that vessel segment. Okay. And so what we've done now is that, when we reach the end of the tree, we're able to compute the impedance of just this vessel segment right here. Okay. Okay. All right. And so now that we've got the base case, now we have to do the recursion case. Okay. All right. And so if we're not at the end of the tree, we're at a case where our vessel here is going to spawn 2 kids. Okay. It's going to have a nice blood vessel family. Okay. And so the first thing that we need to do is we need to compute the input impedance of both of the daughter vessels. And this is going to utilize recursion. Okay. Okay. And so, we want the impedance of the 2 daughters. Okay. And so just like we did with resistance, the way that we're going to compute this is we're going to call the exact same function. Okay. And so the function name here is calculate impedance. Okay. And so this is going to tell MATLAB that we want to calculate the impedance of our daughter vessel. Okay. Right. And remember our rule. And so remember our rule is that, when we create a daughter, then the daughter is going to be 1 order less than our parent vessel right here. Okay. And so when we call the function recursively, we have to call it with order -1. Okay. And so that tells us that the order is 1 less than our parent one right here. Okay. And in this case, we're going to keep the same frequency. And so we're just going to load in the same frequency variable. Okay. And so that's daughter 1. Okay. Favorite daughter. And now we're going to do the other daughter. Okay. The one that gets hand-me-downs. And so we're going to use the exact same function there. It's going to be the exact same thing. So in this case, we have, I guess it's kind of cruel. So I guess we have twin daughters, but I guess we have one daughter that's more favorite than the other one. She will be a higher order than you. You're going to be a good dad. You're going to be a good dad then, right? Okay. All right. So and so that's the only place where recursion comes in. Okay. And so at this point, what you have to do is you kind of have to trust that when you call this function recursively, then it's going to return the exact impedance that you need. Okay. And so what you have to do at this point is, now that you have these 2 daughter vessels, okay, what you have to do here is we have to combine these 2 daughter vessel impedances in parallel. Okay. And so this is exactly the same as what we did for the resistance, because right now we have these 2 daughter vessels, okay, and we want to lump them together into one impedance, and so that we can use it as a terminal impedance for our parent vessel, okay. Right. And so if you read the comment here, then that's what we have to do. So we have to combine these together in parallel to form the terminal impedance of this branch, okay. Okay. And so let's go ahead and impedance, okay, terminal impedance. This is just going to be the same thing as, combining our 2 daughter vessels in parallel. Okay. Right. And so if you have 2, it's the same thing if you have 2 resistors in parallel. And so the formula for that is going to be 1 over terminal impedance is equal to 1 over daughter impedance plus 1 over daughter impedance. Okay. And so we're going to do that, and then we're going to invert this whole thing, and so we're going to raise it to the -1, and that's going to give us our terminal impedance. Okay. Okay. And so now if we go back to the notes, okay, what we have now is we have this. Okay. And so this is exactly what we just computed. We've computed ZIT. Okay. All right. And so now what we can do is we can take this ZIT and then use it to compute a new reflection coefficient for this vessel. Okay. All right. And so if you're given the terminal impedance and you're also given the characteristic impedance, what you can do is you can use those two things to compute the reflection coefficient for this vessel. Okay. All right. And so if we look at this formula here, we can see that lambda or gamma, the reflection coefficient is equal to Z naught minus ZT divided by Z naught plus ZT. Okay. And so we have ZT, and so ZT is this terminal impedance right here. We just computed that. And we have Z naught, okay? And so Z naught is up here, okay? And so let's do that to compute the reflection coefficient, okay? And remember, you know, we're doing this for a case when we're in the not base case, right? Because remember, in the base case, our reflection coefficient is 1, but for all other cases, when the order is greater than 1, you know, we have to compute the reflection coefficient using this formula, okay? All right, and so this is going to be Z naught minus terminal impedance divided by Z naught plus terminal impedance. Okay. All right. So any questions on that? Does that make sense? Why we compute this reflection coefficient for this vessel here? Okay. Okay. And so now that we have this reflection coefficient, now we can use this to compute the input impedance. Okay. So actually we can use the exact same formula that we have here. Okay. Because all this stuff is going to be the same. Right, so I'm going to use, oops, go ahead and copy and paste this here. Okay. Right. Because if we look at this formula, we have Z naught, and then we have the reflection coefficient. Okay. Oh, we have to define L. Actually, let me define L out here. And so we have the new reflection coefficient, which is the one that we computed from Z naught and ZT. Okay. We have lambda 2, which is what we have up here already. We have L, which is what we defined up here. Okay. And now this is going to return the impedance for this vessel here. Okay. Let me just check to make sure. Yes, that is everything. All right. And so that's problem 4 for you, right? So that was more in depth than I planned it to be, but hopefully this has it make more sense for you. Okay. And so if you implement it like this and then you run the other script, you should be able to get the results that I have in the homework document. Okay. Okay. So any questions on this? Yeah, it's tricky, and I think it doesn't help that we did this a couple of weeks ago too, and so you know, glad. Hopefully, this was helpful for you guys. Okay. All right, and so that's the homework for you. Okay, and so now let's go. Now let's move on to the lecture notes that I have for today, which is on microcirculation. Okay, and so let me do this. No, that's not what I wanted. Slideshow. View, reading view. Okay, and so I have to do this, otherwise the chat gets lost. All right. Okay. All right, and so we're continuing kind of on the same theme that we were going over on Tuesday, and we're basically finishing up the biomechanics of the rest of the circulation. Okay, and so on Tuesday what we did was we covered the venous circulation, right? And so we spent a bit of time talking about veins and all the cool stuff that that has to do with that. Okay, and so the last part of the cardiovascular system that we haven't talked about the biomechanics for is the microcirculation. Okay, right. And so here are the learning objectives. And so you know, hopefully we're gonna cover all of this stuff today. Okay? All right, and so let's first thing let's remind ourselves of what is the microcirculation exactly. Okay, and so the microcirculation basically refers to all of the vessels that are really tiny. Okay, and so that's why we call it the microcirculation. Okay? And so the names of these vessels are things like arterioles, things like capillaries, things like venules. Okay, and so all of these are, and so the only place where you see these kinds of vessels are in the organs. Okay, the organs and the muscle. Right. And their primary function is to kind of act as a transport zone, okay, or a transfer zone, I should say, okay. Because remember, the primary function for the cardiovascular system is to transport oxygen and other nutrients to all the parts of your body, okay. And in order for all your muscles and your organs to actually use those things, we need to have a place where your muscles can actually extract the oxygen and the nutrients away from the blood, okay, and all that happens in the capillaries, okay. And so the reason it happens here is because these vessels are extremely small, right? It's so small that you can't see them with just with like a normal cardiovascular imaging, okay. And so if you're looking at kind of the CT scans for your projects, you won't see any capillaries or arterioles because they're too tiny, and so you can't. The image resolution is just way too small, or just not good enough to see these vessels. Okay. All right, questions. How does this relate to asthma? That's a good question. So asthma is more of a lung disease, as I kind of understand it, and so I'm not actually sure, but I'm sure it does have some effects on the capillaries inside your lungs. But I don't really know enough to comment on that. But I'm sure it's related, but I'm not sure exactly how much. Yeah. Okay. All right. And so because they're so small, they're so small that it's a blood vessel. They're basically the same diameter as a blood vessel. Okay. But another consequence of them being so small is that you lose a lot of energy trying to squeeze through these vessels, right? And so if you can imagine, if we kind of look at our law of resistance, right? We know that the viscous resistance of the vessel dramatically increases when the vessel radius goes down. Okay, and so since these vessels are kind of the smallest that you can get, the viscous resistance for these is extremely high. Okay. And so if you think of like our Ohm's law rule, which relates the pressure drop to the flow, it's going to take a lot of pressure drop in order to get blood flow going through these because they're so small. Okay, and so these kind of act as kind of a separation zone. Okay, because if you remember what we talked about on Tuesday, where we said that flow in your arteries is very high pressure, very high flow, but flow in your veins is very low pressure. Okay. The reason the veins are in such low pressure is because of the capillaries. Okay? It's because the blood loses just so much pressure trying to go through the capillaries that there's just not much left for it when it gets to the veins. Okay. And another important characteristic of the microcirculation, specifically for the arterioles, okay? And so you can think of the arterioles as kind of the very smallest arteries that you have, is that these arterioles are actually very muscular, and so they're lined with these smooth vessels that allow them to really change their diameter very rapidly. Okay, and so this is actually a key component of the microcirculation that we'll talk about, in later parts of the class. Okay. Okay. So any questions so far on the microcirculation? Oops. Okay. All right, and so let's talk about the specific functions, okay? And so we talked about what the microcirculation is. Now let's talk about its physiology, okay? All right, and so the first function that it serves, and this is a very fluid mechanics thing, is that they serve as the primary resistance to flow, okay? And so because the arterioles can really control their diameter, they can really become as tiny or as big as they need to be. Okay, and so these arterioles really dominate the amount of viscous resistance that you have, overall in your cardiovascular system. Okay, and so these arterioles, they're really the ones that are holding all the controls for, what the blood pressure in your system is going to be and what the flow rate is going to be. Okay, because depending on how much the arterioles dilate or how much they constrict, that really can have a big impact on what the pressure and flow is in your cardiovascular system. Okay. And so if you think about, if you think about your synovascular models, a lot of you guys are kind of moving on to the simulation stage at this point. For your outlet boundary conditions, we're supplying a resistance boundary condition, right? And so, that resistance is meant to model, the viscous resistance of the arterioles and the capillaries here, right? Okay, and so the next function of the microcirculation, kind of like we talked about on the previous slide, is molecular exchange, right? And so, because these vessels are so small, they're basically the size of the red blood cell. So as the red blood cell basically flows through a capillary, the walls of the capillary are in contact with the red blood cell, and so it really makes it easy for kind of the red blood cell to kind of drop off its contents, right? And so if you think of the red blood cells as kind of like a mail carrier, right? And so the capillaries are basically like the mailbox, okay? And so the mailbox basically allows the red blood cell to just drop off whatever it's carrying to the muscle or the organ that it's going to. All right, and so another feature of the microcirculation that we're not going to talk about too much is the fact that it allows this thing called flow bypass. Okay, and so if you look at this diagram here, you'll see that there's kind of these vessels here called the arteriovenous bypass. Okay, and so what these allow, and so the function of these things is to allow blood to basically bypass the capillaries. Okay. Because it could be, depending on what's going on with the rest of your body, maybe, your body wants the blood to kind of bypass that and not have to squeeze through the capillaries, okay? Because maybe the body has to route that blood somewhere else, maybe say like, you cut your hand or something like that, okay? And if your body wants to direct blood away from your feet in order to go to your hands to heal that wound, the microcirculation actually has a mechanism to do this, right? And so that's one of the nice, cool things about the microcirculation that's not talked about that often, actually. And so it's kind of a cool thing. Okay. Right. And so the last function of the microcirculation that we'll talk about is capacitance. Right. And so just like the veins, the capillaries and the microcirculation can store some blood. Okay. And so it's not nearly as much because the microcirculation is so small. But it's estimated about 22% of the total blood volume is stored in the microcirculation at any given time. Okay, right, and again that's due to the fact that the vessels can expand and kind of store flow in it. Okay. Okay. So any questions on this so far? Okay. All right, and so let's look at a plot to see of how the velocity and the pressure, varies as you go through the microcirculation. Okay. All right, and so the way to kind of read this chart is, we're going to read it left to right. Okay, and because remember the blood always starts in the arterial system and then ends up in the venous one before it ends up, going back to your heart. Okay, and so the first curve I want you to look at here is the top one, which shows the pressure. Okay. And so you can see here as the blood flows from the arterial to the venous system and it passes through the microcirculation, you can see here that the pressure goes down. Okay? And in fact, you see a very rapid decrease in pressure as you kind of go through the arterioles, which is right here. Okay. And remember, so this pressure drop is a result of the fact that your microcirculation is so small and you have a very large amount of viscous resistance going through those microcirculation. Okay. Okay. And so that's the pressure, okay? And then the other thing is the velocity, okay? So the velocity does something interesting where it goes down and then it comes back up, right? And so that's kind of a little strange because, we know that the pressure is going down. And so why does the velocity come back up? And the reason for that is actually related to your previous homework, right? And so, what's not really shown here is that, as the vessel goes as the blood flows from the arterioles to the capillaries, what's happening is that, there's a ton of bifurcation going on, right? And so you might have one arteriole and you might have like 7 or 8 capillaries that branch off that, right? And so, the arteriole is like octomom basically, okay? And because the, because those arterioles are branching out so much and they're having so many kids, right? The flow is basically splitting among all these different capillaries. Okay, and so even though each individual capillary is a lot smaller than each individual arteriole, because there's so many capillaries that are out there, the velocity actually ends up going down. Okay, because if you look at kind of each individual capillary, because each individual capillary is only taking a small fraction of the flow from the arterial, the velocity actually goes down because there's just a lot less blood. Okay, and then when the capillaries kind of reconverge up in the venules, okay, and so each of those, maybe like hundreds and hundreds of capillaries, they might come back and form maybe like 5 or 6 or 7 different venules. Okay, then because you have so many capillaries converging on the same vessel, then the velocity has to go up because you have so much blood that's converging into one place. Okay. And so that's kind of the explanation of why the velocity kind of goes down first and then it comes back up here. Okay. All right. So any questions on this? What is mesenteric cat? I think this experiment here was done in a cat. And so I'm not sure what kind of what mesentery means, but yeah, usually these kinds of very detailed measurements, they either weren't done in humans or they were done in humans and they were done by Russians, because Russians, they have very different ethical standards for human subject research, and so yeah, it'd be very hard to get this kind of data in humans, and so yeah, a lot of these kinds of data is done from animal studies. Yeah, I'm not throwing shade. I mean, it's good. It's good that otherwise, we actually use a lot of their data for a lot of stuff. It's just easier to do that when you don't have research ethics. Yeah. All right. Yeah, how is this physically better? Yeah, it's a good question, because you're thinking even in a cat, you're talking about these vessels are extremely small. So I'm actually not familiar with this particular study and how they do that. They might do it via MRI, and so MRI I think is probably a little bit more likely because ultrasound is a little bit too low resolution for this. And so they might use a very high-powered MRI machine to measure these things. But for this one, I'm not exactly sure. And so I found this figure in the textbook, and then I just kind of used it. But I'm not exactly sure what they used. Yeah. Okay. And so let's talk about the blood flow in these microcirculations. Okay. All right. And so because these vessels are so small, the blood cells themselves are now all of a sudden going to become much more prominent. Okay, and so if we think about the size of a typical red blood cell, and so if you think of it like an oval, then the dimensions of that oval are about 7.5 micrometers by 2 micrometers. Okay, usually this is much smaller than most vessels that you have in your body, and so most of the time we just ignore their presence, right? And so if you remember kind of from the beginning of class, this is kind of the whole debate that we had about, whether blood is a Newtonian fluid or not, okay? But when you're talking about kind of the larger arteries and the larger veins, you can consider it as a Newtonian fluid because the blood cells are so small, okay? But, when the blood reaches the microcirculation, the diameters are so much smaller, they're basically on the same order of magnitude as these blood cells, okay? And so we can't ignore their presence anymore. And so, once we start to look at these effects of these particles in this flow, they create some actually really interesting and complex effects, right? And so the first of which is called cell migration and depletion. Okay, and so what that means is that whenever you have particles that are in a suspension and you flow that into a circular tube, then what tends to happen is that all the particles are pushed. They're pushed away from the edges of the tube, and they're pushed away from the center. Okay, and so what ends up happening is that if you have your vessel that looks like this, you kind of have a donut of particles that kind of ends up forming. Okay, because what happens is that basically the particles are going to collide with the walls of the container. Okay, and so that's going to push it away from the walls. And they're also going to collide with each other in the center. And so that kind of pushes them away from the center of the cell too, of the vessel. And so you have kind of like a donut effect of like, if you look at kind of a circular cross section, then, kind of a donut on the interior, that's where most of the particles are going to be. Okay, it's a very interesting effect, and I actually had a colleague in grad school that is actually studying this as well. And so and I'm going to butcher this name as I always do. And so this is called the Segre-Silverberg effect. Okay, and so if you look at videos of this, it's very cool. And so it's a very cool effect, and blood cells kind of follow the same the same behavior. Right, and so that's one interesting effect that happens when you add these particles into the flow, and they're about the same size as the vessel. And the other effect that happens is our viscosity tends to change. Okay, and so because the red blood cells, they migrate away from the vessel walls, they change the effective viscosity in these vessels up to a 5 times difference. Okay. And so that's a huge difference. And so, when you're considering the viscosity of the flow in the microcirculation, you can't ignore the particles because they have a huge effect on the viscosity. Okay, and this is one of the primary complications of simulating flow in the microcirculation, because there's not that many great models out there for how to model the variable viscosity. Okay, there's some that people use. But we've yet to find kind of a really good model for it. Okay. All right. So any questions on this? All right. Question: is the viscosity high enough to implement creeping flow? That's a great question. And so actually, what tends to happen for these is that the viscosity actually goes down. And so I say a 5 times difference, but it's actually 5 times less viscosity than normal. But in these situations, you do have creeping flow. But the creeping flow kind of comes more as a result of the really small size, right? And so remember, and so this is kind of an aside for those of you who had me last year for advanced viscous flow, EGME 520. And so in order to get creeping flow, you have to have a Reynolds number that's much less than 1. And so that you actually achieve that in the microcirculation, but not because of the viscosity, but because the diameters of these vessels are just so tiny. And the velocity just end up being really low too. And so even though the viscosity goes down, the diameter also goes down, and also the velocity also goes down so much that you end up with Reynolds numbers that are like in the decimals, like 0.00 something. Yeah, so you do have creeping flow, but it has less to do with viscosity and more to do with size. Yeah. Okay, and so let's go ahead and move on. Okay, and now let's talk about the, oh, I went through two things, okay? Okay, and so the interesting thing is that, because the blood cells tend to shy away from the walls of the vessel, you could have an effect like this called plasma skimming, okay? And so if you have a vessel that bifurcates and it's next to the wall, and the geometry is kind of, kind of strange enough, what can happen sometimes is that this side branch right here might not get any red blood cells at all. Okay, and so basically what you can think of this as if you have this blood vessel that's branching off, the fluid that's coming into this vessel is strictly just coming from the area that's right next to the wall. Okay, and because there's no blood cells that are next to the wall, then you might end up with a capillary that gets no blood cells at all, right? And so this is an effect known as plasma skimming. Okay, right. And so that's something that's interesting, because that basically tells you that you have some capillaries in your body that are basically doing almost nothing because they're not getting any blood cells, and so there's nothing there for them to exchange. Okay, all right. And so another effect that can occur is you might have what's called reduced and variable hematocrit. Okay, and so hematocrit, if you remember from the beginning of the class, this defines the red blood cell by concentration. Okay, and so this is reduced in the microcirculation because of this kind of cell aggregation effect right here. Okay, and so you could have hematocrit as low as 25% compared to say like 40 or 50% for kind of the larger arteries. Okay, and so these two things are kind of as a result of the same thing, where the blood cells kind of tend to move away from the walls. All right, question. So does this have to do with flow towards the path of least resistance? You can kind of think of it like that a little bit. And so, it's, the blood cells, in order for it to come to get sucked into the side branch right here, there has to be kind of a significant kind of sucking force that kind of that pulls it in there. But because of the fluid mechanics, there is a lot less resistance, and it is a lot easier for the blood cells to kind of just continue along its way. Yeah. And does Darwinism play into this? What do you mean by Darwinism? Like survival of the fittest?
Yeah, I was just wondering if like will that vessel die, or will it like become bigger using mechanotransduction, just like become bigger so that way flow can actually flow? Oh
yeah, no, that's a great question. I'm actually not sure. And so that would be something I would have to look up. But I would imagine that if a capillary did form like this, and the organ wasn't getting any nutrients from it, then I would imagine that the body would either adapt it so that it might make this bigger, or it just might form just a whole new branch in order to fix it. But the exact mechanism for that and how often that happens, that I'm not sure. Yeah. Okay. All right, and so let's talk about the myogenic effect and the BAYLISS effect, right? And so the myogenic effect, remember I mentioned at the very beginning of this lecture that the arterioles, which are part of the microcirculation, they're very muscular, and so they're lined with smooth muscle, right? And so they have they can control their radii very, very strongly. Okay. And so one way in which they control their radii is through the myogenic effect. Okay, and it's basically a way for the microcirculation to maintain or to manage their diameter in response to changes in pressure. Okay, and so the way it works is like this. And so these three graphs kind of work in tandem. And if you think of like mechanotransduction, this is very similar to that. Okay, and so the first graph to read here is the first one. Okay, and so for the first one, let's say that we have an increase in arterial pressure. Okay, and so let's say that you decided to go run 2 miles. Okay. And so when you're exercising, your blood has to pump a lot harder, and so your blood pressure is going to go up. Okay, and so you can kind of think of this as the point where you start running and you realize you're out of shape, and you're like, and your blood pressure is doing crazy. Okay, right. And so the first thing that happens when your blood pressure goes up is that the pressure in your microcirculation is also going to go up, right? And so you can see here that as a result, in your capillaries, your blood pressure is going to go up in direct response with the arterial pressure. Okay, and so in response, the right away, your arterioles, which are your smooth muscle, they're going to increase their radius because there's more pressure on them. There's more pressure on them that's going to push them out basically. Okay, all right. And so, this increase in pressure is something that the microcirculation doesn't like. Okay, and so in order to basically counteract this, what the microcirculation is going to do is it's going to contract. Okay, and so as it contracts, then the blood pressure is going to start to stabilize within the capillaries. Okay, right. And so this effect right here, where the radius, it goes from a maximum here when the blood pressure initially goes up, down to a minimum after the smooth muscles contract, this is called the BAYLISS effect. Okay, and so this was first observed in 1903, and then later on, I think the theory was kind of developed more, and it became what's known as the myogenic effect. Okay, and just like all things, it was named after some dude named BAYLISS. I don't know who he is, but that's how it was named. Okay. All right, and so the purpose of this effect is to maintain the capillary pressure within a small range, okay? Because the capillaries, remember one of their main functions here is to basically be a place of exchange between the blood and your organs, okay? And so that has to be a very tightly controlled process where the pressure can't go up too much and the flow can't go up too much. Otherwise, that exchange, it does not really happen properly. Okay, and so the myogenic effect, the body kind of developed this as a response to basically ensure that the exchange takes place, as efficiently as possible. Okay. Okay, so any questions on this? Okay, all right, and so let's talk about the vessel wall mechanics. Okay, all right, and so similar to the venous system, you know, the microcirculation, you know, because they're very tightly packed into your organs, they face a lot of external pressure as well. Okay, and so this is going to cause some effects on the cross-sectional area of the vessels. Okay, right. And so just to kind of remind you from Tuesday, you know, the difference between the external pressure and the internal pressure of a vessel is known as the transmural pressure. Okay. And remember, you know, in the same way with the veins, you know, we have this transmural pressure not because these vessels are being squeezed so tightly, right? It's not that your muscles are squeezing these super tight. It's more that, you know, by the time the blood reaches the microcirculation, the blood pressure internally on the inside of the blood of the vessel has dropped just so significantly, just due to the viscous resistance. Okay, right. And so if you plot the relationship in between the cross-sectional area and the transmural pressure, you know, in the vessels, you can see that it's generally nonlinear. Okay, right. And so that's what you see right here. And so it's not like a straightforward stress strain curve, and you have this kind of like you know, square root curve that's like this. Okay. And if the vessel has smooth muscle like the arterioles, you know, this can affect this graph, you know, even more, because the smooth muscles are going to kind of push back on the transmural pressure. Okay. Okay. So any questions on this so far? Working out their calves, yeah. You got to get those calf muscle pump going so you get your blood back to your heart efficiently. Okay. All right, and so here's a couple more graphs where you can see. And so you can see here that if you actually plot out the stress strain curve for the microcirculation, you can see here it's very nonlinear. And so you can see here we have either a parabolic or even exponential graph here. Okay. And you can see here that, you know, when you do have smooth muscle in your microcirculation, then you can actually, you know, decrease the vessel diameter when the blood pressure goes up. Right? And this is the myogenic effect that we talked about before. Okay. And so when you see the term active here, what that means that there's some active control due to some muscles that can, you know, that can affect the diameter of your vessel. Okay. And so now let's talk about pulsatility. Okay. And so if you kind of remember from our lectures on, you know, oscillating flow in rigid vessels, remember we have this non-dimensional number known as the Walmersley number. Okay. And so for the microcirculation, the Walmersley number is actually really low. And so what that tells us is that, you know, there's very little pulsatile effects that take place in the microcirculation. Okay. In fact, you know, because of the tight control that's given by the arterioles, the flow is almost constant in the microcirculation, and so you almost have kind of a steady stream of flow, which again, you know, the purpose of that is to make sure that the blood cells can do its exchange and can do it, you know, efficiently. Okay. But with that said, you know, it is possible for the microcirculation flow to be a little bit pulsatile. Okay. And so the reason for that, and so there could be some reasons for this. And so the first reason for this is it could be that or it's just from the fact that your arterial pressure oscillates, right? And so if you think about your blood pressure in your arterial system, it goes kind of up and down just naturally. Okay. And so some of this pulsatility, you know, it just naturally reaches the microcirculation. And so even though most of these is damped out, some of that pulsatility still happens in the microcirculation. Okay. So you can't get rid of all of it. Right, and so you have a little bit of pulsatility also from the myogenic effect. Okay, and so the fact that your arterioles are kind of contracting on themselves in response to the pressure, this also causes some pulsatility in the microcirculation flow. Okay, and you also get some pulsatility just due to the fact from tissue compression. Okay, and so when the tissue around your microcirculation is compressed, this can also cause the flow in the microcirculation to be pulsatile as well. Okay, right. And so, you know, generally speaking, you know, relative to the arterial system, flow in your microcirculation is very constant. Okay, so you have a very constant stream, but there could be some pulsatility and there could be some oscillations that take place. And the reason is because of these right here. Okay. Okay. Any questions on this? Okay. All right, and so let's talk about wall shear stress. Okay, and so just like other parts of the cardiovascular system, wall shear stress plays a critical role in regulating the size of the microcirculation. Okay, all right. And so if you compare the wall shear stress values on the arterial side and the venous side, wall shear stress tends to be a little bit higher on the arterial side of the microcirculation. Okay, and so again, this is kind of due to the fact that the arterial side has higher flow rates and higher pressures and higher velocities. Okay? And so that tends to give you higher wall shear stress values. Okay. Right. And so that kind of that's kind of illustrated here as well. Okay. And so I think this is the same cat as before. Okay. And so here they're measuring the wall shear stress on the arterial side of the microcirculation and the venous side. And so you can see here that the arterial side has higher wall shear stress values than the venous side. Okay. Right. And kind of going back to your question from earlier, Josh, the microcirculation also has the ability to form new blood vessels, and kind of the primary trigger for this is the wall shear stress. Okay, right. And this is actually a good thing. And so and this is kind of a reason why people tell you to exercise regularly. Okay, because if the if you exercise regularly and you kind of put your microcirculation under a lot of stress, on a regular basis, then what your body's going to do is going to say, hey, Josh or Justin, they exercise a lot, right? And so we should form some more micro vessels in order so that our muscles here can get more oxygen more efficiently. Okay, and so I think I've heard this in commercials before. They said if you exercise regularly, then new blood vessels will form. Okay, and that kind of sounds bogus. I think the first time that you hear it. But it's actually true, right? And so if you exercise on a regular basis, then your body's going to create a lot more of these capillaries and a lot more of these microcirculation, so that your muscles can get your muscles and your organs can get oxygen a lot more efficiently. Okay, but if you kind of sit on your butt all day and you kind of live a sedentary lifestyle, then your body's like, Justin's not really doing much with his life, and so he doesn't really need any capillaries, and so let's just do just the bare minimum just to get by. Okay. Which when you're young is all fine and good, but as you get older and you're gonna wish that you had more capillaries in your body, because that's going to really affect your long-term health. Okay. Yeah, that's true. It's not a good class to take after a year of sitting around in COVID. That's for sure. Yeah. Yeah, it's funny, when a lot of this research, just naturally has to do with cardiovascular health and stuff like that. And so when you're doing this kind of research as like I mean, engineering students, as we all know, we're all really busy, and so if you're learning a lot about this stuff as an engineering student, when you're on your like third cup of coffee and your third cup ramen for the day, it's not the best thing for your mental health. It's like, oh man, I'm eating my third cup ramen today. I'm going to get really high blood pressure, and now I understand exactly what that's going to do to my body. And so it's not a comforting thought. That's for sure. Yeah. And so this process through which your body creates new blood vessels to kind of meet the demands of your body is known as angiogenesis. Okay. And so angio is kind of that's a kind of a prefix here to denote to denote anything that has to do with blood. Okay, and genesis is a word that has to do with creating new stuff. Okay, and so this is a word that basically means creating new things in your cardiovascular system. Okay, and it's something that you want. And so definitely I know now is not a good time because it's really busy, but after the semester definitely get out there and exercise again, because you want this stuff. Yeah. Yes, so angiogenesis is the formation of new blood vessels. Sounds like something Sephiroth would use. Yeah. Yeah. Sephiroth used angiogenesis on you, so you use Knights of the Round to counter him. Okay, so any questions on this? Okay. All right, and so what's this graph right here? Let me see. Okay, wall shear stress, blah 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 blah, function diameter and pressure. Okay, so this is just more data that kind of reinforces the points from the previous slide. Okay, And so as the diameter goes down, the wall shear stress also goes down. And then as the pressure goes up, then the wall shear stress also goes up. Because as pressure goes up, that's going to force more blood flow into your vessels. Okay. All right. And so this graph right here, on the left, basically shows you kind of the values of wall shear stress that your vessel typically likes for certain diameters. Okay. And so just like the rest of your body, your microcirculation is going to want to try to maintain the wall shear stress for those vessels of those diameters. Okay. And so we spent a lot of time talking about the fluid mechanics of microcirculation. Now let's talk a bit more about the molecular transport, because this is kind of the key function for the microcirculation. Okay. And so just to kind of remind you, remember the key responsibility of the microcirculation is to kind of be a place where oxygen and nutrients can be exchanged between your organs and your blood cells. Okay, right? And the primary mechanism or the primary physical way that that transport occurs is through diffusion. Okay, and so if you never heard the term diffusion before, the definition of diffusion is basically the passive transport of a material through a medium. Okay, and so the example that I always give in all my classes is, say, let's say that you're in your room, okay, and you have an air freshener, and then you just spray the air freshener on this side of the room. Okay, if you then go run on the other side of the room, eventually that air freshener is gonna come to you, right? And so eventually you're gonna smell it, even though there's no airflow in the room, and so there's no fan or anything that's pushing the the air freshener particles. Eventually that air freshener kind of spreads out and kind of goes to the rest of the room. Okay, and that action, and so that kind of transport of material is known as diffusion. Okay, and so I'll add a little bit more to the definition for this one in particular. And so one thing that's needed for diffusion is that you need to have a gradient of concentration. Okay. And so things typically flow, or things typically diffuse from high concentration to low concentration. Okay, and so the way that works for a blood vessel is that what tends to happen is that your blood cell here is rich in oxygen, and so it has kind of a full load of oxygen that it's carrying. Okay, and so if you compare that to kind of the surrounding tissue around it, it might not have that much oxygen, right? And so if you compare here within the blood cell, we have high amount of oxygen. And then on the other side of the vessel wall, we have a low amount of oxygen. Okay, and so because we have this gradient where we have high oxygen and low oxygen, the oxygen is going to want to naturally just go to the place where it's there's lower oxygen. Okay, and that's called diffusion. Okay, right. And so that's the primary mechanism through which oxygen molecules get transported through your blood vessels. Okay. All right, but there's a limit to diffusion, okay? And so diffusion, you can only go so far through diffusion because it's kind of a passive process. And so the way that we kind of characterize this is we say that oxygen typically has a diffusion length of about 20 to 200 micrometers, okay? And so once the oxygen has traveled that far, it can't really go anymore, because that's kind of the limits of diffusion, okay? And so in order to basically make sure that all your body parts can get the oxygen that it needs, it needs to create this kind of vast capillary network. Okay, and so the capillary network kind of spreads out all throughout your organs. Okay, and it's designed in such a way that most tissue in your body are within about 100 micrometers of a capillary. Okay, and so if you take any kind of random organ and any kind of random tissue in your body, and you go 100 micrometers in any direction, eventually you're going to hit a capillary. Okay, and that's basically to ensure that every single organ gets the oxygen that it needs, because otherwise if you have organs that are really far from a capillary, it's not going to be able to get the oxygen and nutrients that it needs, because it's so far from the place where the transport takes place. Okay, and so the body kind of naturally figures this out on its own, and so it naturally just knows that if we have an organ and it's something that's going to need oxygen and nutrients, we need to put a capillary there in order to get there. Okay. Right. And so you can kind of almost think of this, and so kind of a good analogy that I like to use for this is freeways, right? And so freeways, you can kind of think of them as the capillaries, because that's where the blood vessels go. Okay. And then each of the cars on the freeways are kind of like the same things as your blood cells, okay? And so in order to make sure that, you know, each car is able to reach its destination within an efficient amount of time, you know, the city planners a long time ago, they designed the freeway such that they run through, you know, key parts of neighborhoods, okay? And they design, you know, each of the freeways such that, you know, if you need to get to a certain neighborhood, you know, it's always within a certain amount of distance to one kind of freeway exit, okay? And so capillaries are basically the exact same thing. It's a car crash, a blood clot. Yeah, a car crash is basically a blood clot. Yeah. And so if LA was a human, LA would die every day because it has blood clots on its freeways every day, and it's a mess to go through. Yeah. All right. So any questions on this? Okay. All right. So what other slides do I have here? Okay. Let's see. Okay, yes, and so another determining factor that determines, you know, how much of a substance passes through a capillary wall is pressure difference, right? And so if you can imagine, you know, pressure is nothing more than just a force that's distributed over over an area, right? And so if you have a higher pressure on one side, you know, that's going to push, and a lower pressure on the other side, that's going to push more stuff from the higher pressure location to the lower one, okay? And so pressure differences also play a key role in helping to encourage capillary transport, okay? And so there's actually two contributions to the pressure difference, right? And so the first contribution is just hydrostatic pressure. And so this is just differences in fluid pressure as we kind of know, right? And so you have a high fluid pressure on the inside and a low fluid pressure on the outside, okay? And so this is going to naturally push some nutrients through and oxygen through that pressure difference, okay? And so that's hydrostatic pressure, okay? Another source of pressure difference is called the osmotic pressure, okay? And so what this is a little bit more difficult to understand, but basically what happens is that if you have kind of a place, if you have kind of a fluid with a lower volume or a lower water content, okay? Then what happens is that, you know, water is going to tend to flow from the place with higher water content to lower water content, okay? And this has nothing to do with hydrostatic pressure, right? And so just as an analogy, you know, if you have, let's say that you have two cups, right? And they're both equally full. But on one cup you have, let's say you have salt water, okay? And so you've collected that water from the ocean, and so it's very heavily salty, okay? And so on the other cup, what you have is just pure water. And so maybe you took it from your Brita filter, or maybe even just from your faucet, okay? And so if you compare the two cups, you know, the volume of liquid in there is the same, but the salt water is going to have more salt by volume and less water by volume, okay? And so if you connect the two cups together, what's going to happen is water is actually going to flow from the freshwater cup to the saltwater cup, because the saltwater cup has less water there by volume. Right. And so the same thing actually happens in your capillaries too, okay? Because in your capillaries, you know, the blood cells and the platelets and all that stuff, they take up a significant portion of the volume, okay? But maybe outside of the capillaries, you know, you don't have those cells, and so they have less, you know, those things, okay? And so there's always going to be a net osmotic difference, which is actually going to push fluid. It actually tends to push fluid into your capillaries because the blood in your capillaries is not that much actually liquid by volume. Okay, and so, you know, both of these pressure differences also help to enhance, you know, a molecular transport. Okay. Okay. So any questions on this? Okay, and so we're almost out of time, and so let's talk about just the last function for the microcirculation, and that's flow control. Okay. And so remember, you know, what we talked about earlier, that the arterioles, they can really constrict their diameters quite a bit, okay? And so if they restrict their diameters a lot, you know, that increases their resistance quite a lot, okay? And so, of course, this is going to change the total amount of resistance in your body, okay? And so if you have a higher resistance, then that's going to decrease the amount of blood flow that goes in your body, okay? And so the arterioles actually have a lot of control over how much blood actually goes through your body at any given time. Okay. And so the factors that that tell the microcirculation or that tell the arterioles to whether to dilate or to constrict, there's three kind of determining factors. Okay. And so the first is metabolic control. Okay. And so metabolic control refers to hormones in your body. Okay. And so your body can release hormones that basically tell the arterioles that, hey, we need to increase the amount of flow that's going here because we need more demand for the muscles. Okay. And so once the arterioles kind of sense those hormones, then they can kind of expand and allow more flow. Okay. All right. Another form of control comes from shear stress. Okay. And so just like as what we discussed in mechanotransduction, where these blood vessels, they're going to respond to changes in shear stress and change the diameter. Okay. And the last part of control is myogenic, right? And so that's kind of the effect that we talked earlier, where the diameter of the microcirculation can respond to changes in pressure. Okay. All right. And so that's the end of the lecture notes on microcirculation. So any final questions before we wrap up for today? Okay. All right.
Hi, Professor.
Oh, your question?
May I have the ID for the geometry?
Oh, yes. Let's talk after class about that. Yes.
Okay.
Okay. Okay. Yeah. So thank you everyone for tuning in today. Remember, our midterm is a week from today, and so actually later tonight I'm going to post a poll for you to vote on the topics that you feel most uncomfortable with, and then tomorrow afternoon I'm going to record a review session for you to review those topics. Okay. All right. And so thank you guys for tuning in today. I hope you guys enjoy the rest of your evening, and I'll see you guys next week.
Beautiful. Thank you, Professor.
Thanks, guys.
Thanks, Professor Tran.
Thanks, guys. All right, Steven, so you need the ID number for the for your project?